In this mini lecture we will discuss environmental health and hazards, types of toxic substances and their toxicity, and the study of toxic substances and their effects. After reading chapter 14 and watching this video or reading the transcript that goes with this video, you should be able to explain the goals of environmental health and identify major environmental hazards, describe the types of substances in the environment, the factors that affect their toxicity and defenses that organisms possess against them, discuss the study of hazards and their effects, including wildlife toxicology, case histories, epidemiology, animal testing, and dose-response analysis. Environmental health is the science that assesses environmental factors that influence human health and quality of life. Both natural and human-caused factors influence human health, as can be seen here. There are 4 types of environmental hazards that influence environmental health. 1. Physical hazards are hazards that occur naturally in our environment. Examples included UV radiation, earthquakes, volcanoes, fires, floods, landslides, hurricanes, and droughts. Emergency planning helps us to prepare for some of these hazards, while preventative actions, such as avoiding deforesting slopes, which prevents landslides, avoiding channelizing rivers, which prevents flooding, and applying sunscreen. 2. Chemical hazards are caused by synthetic chemicals such as pharmaceuticals, disinfectants, pesticides, harmful natural chemicals, hydrocarbons, asbestos, and lead. We can reduce our risk to these through avoidance, by choosing green cleaners or pesticides, and by staying informed about chemicals you're exposed to. 3. Biological hazards are hazards resulting from ecological interactions. For example, exposure to insects. Biological hazards include infectious disease, which is when viruses, bacteria, and other pathogens parasitize humans and causes disease such as malaria, cholera, and West Nile virus. 4. Cultural hazards are the result from where we live, our socioeconomic status, occupation, or behavioral choices. Smoking is an example of a cultural hazard. Disease is a major focus of environmental health. Despite our technology, disease kills most of us. Over half of world's deaths are from non-infectious diseases or conditions such as cancer and heart disease. Factors that influence incidence of these diseases include genetics, lifestyle, environmental exposure, poverty, and poor hygiene. Infectious kill 14 million people a year. Developed nations have far fewer infection disease deaths than developing nations. Specifically, infectious diseases account for 50% of deaths in poorer nations. Nutrition, sanitation, hygiene, and access to medicine all play a role in the spread of deaths caused by infectious diseases. Social and environmental factors influence the spread of infectious diseases. Our global society makes it easier for diseases to spread across the world. This is further complicated by the fact that some diseases, like influenza, are caused by viruses that evolve rapidly and gives rise to new strains. With a global population, the spread of some diseases can be very dangerous because they could result in a global outbreak of disease, such as what happened with SARS in 2003, H5N1 bird flu in 2004, and H1N1 swine flu in 2009 and 2010. Public health has decreased some infectious diseases, however others, such as AIDS, are spreading globally. Public health is the science that promotes and protects the health of people in the communities where they live, work, learn and play by promoting healthy lifestyles and preventing diseases and injuries. Further complicating public health is the fact that some diseases, such as malaria and tuberculosis, have developed resistance to antibiotics. Another environmental factor influencing the spread of disease is global climate change. As climates warm, this enables tropical diseases, e.g. malaria, dengue, and cholera, to spread to temperate regions. While public health is the science of preventing disease and injury in individuals and human populations, toxicology is the study of the effects of poisonous substances on humans and other organisms. Paracelsus, who lived from 1439 to 1541, is considered to be the father of toxicology. He said, All substances are poisons. There are no which is not a poison. The right dose differentiates between a poison and a remedy. Or in short, the dose makes the poison. This gets to the idea of toxicity, which is the degree of harm a chemical can inflict. Toxicity depends on the combined effect of the chemical and its quantity. Environmental toxicology deals with toxic substances that come from or are discharged into the environment. Environmental toxicology studies use animals and plants as models to see if chemical may be a threat to human health. I was trained to know the difference between a toxic ant and a toxin, and as such it's important for my students to receive the same training. A toxic ant is a synthetic or man-made poison. Examples include pesticides, herbicides, and solvents. A toxin is a poison from biological or other natural sources. Examples include snake venom, mercury, and arsenic. The slide includes Table 14.1 from your textbook, which shows the different types of environmental hazards you can be exposed to. You should take note of these and be informed citizens and consumers and watch out so that you are not exposed to high concentrations of these chemicals. You should be aware that toxic chemicals are all around us. They also exist naturally and in our food in some cases. Don't assume all natural chemicals are all healthy and synthetic ones are all harmful. This is not always true. Plants, even crops, can sometimes contain harmful chemicals. For example, some plants produce toxic chemicals to ward off being eaten. Meat can contain toxins that animals ingested from the plants and animals they ate. Both natural and synthetic chemicals are in our environment. Chemicals can be found in the air, soil and water. What may be alarming to you is that every one of us carries traces of hundreds of industrial chemicals in our bodies. Furthermore, babies are born with chemicals already in their bodies, a quality known as being pre-polluted. Children are especially susceptible to the risks associated with exposure to harmful chemicals because their consumption to body size ratio is far higher than adults. This is why chemicals are said to have a higher body burden on infants and children. Essentially, the same dose of a chemical will have a greater effect and impact on a child than an adult. This is why if only one person or group of people in your house eats organic, pesticide-free food, it should be the children. Not all synthetic chemicals pose health risks, however few of the hundreds of thousands of chemicals on the market have been tested. We will discuss this further in Part 2 of this chapter. Organisms have been exposed to natural toxic ants for millions of years and have subsequently evolved natural defenses to some chemicals. For example, mercury and arsenic are found naturally in the environment and some organisms produce toxins to avoid capture. Such toxins include snake venom, toxins in sea urchins, and pyrethrin in chrysanthemums. Skin, scales, and feathers act as barriers that prevent toxin intake, but toxins can enter into organism through food, water, air. Many organisms have biochemical pathways can detoxify, or in other words metabolize, toxins. When they do this they make the toxins water-soluble so they can be excreted. The liver is a site of detoxification, so it is disproportionately affected by intake of harmful substances. Unfortunately, some toxins can't be detoxified in the body. Those that can't be detoxified are sometimes stored in the fatty tissues and cell membranes of the bodies of organism, including us. Examples of compounds stored in the fatty tissues include heavy metals, dioxins, and some insecticides, such as DDT. Even if an organism does have a detoxification system for a particular toxin, defenses can be overwhelmed if levels get too high. Other toxic ants are harmful at any level. Organisms have no natural defense against synthetics and effects can be severe and unpredictable, which is why we must be careful when we develop new chemicals. Individual responses to hazards vary. Different individuals respond differently to hazards. Genetics can affect responses. People in poor health are more sensitive. Sensitivity also varies with sex, age, and weight. Fetuses, infants, and young children are more sensitive. The Environmental Protection Agency (EPA) sets human exposure standards for adults and extrapolate down for fetuses, children, and infants. However, standards may not be low enough to protect the young. The type of exposure affects the response of an organism. Acute exposures are high exposure to a hazard for short periods of time. Acute reactions usually occur quickly following exposure, are easy to recognize, and follow discrete events, such as ingestion of something, an oil spills, or a nuclear accident. Toxicity tests reflect effects of acute toxicity. Chronic exposures, on the other hand, are low exposures over a long period of time. The response to chronic exposures is delayed, and as the effects from chronic exposures are harder to detect and diagnose. Chronic exposures usually affect organs gradually, such as causing lung cancer or liver damage. With chronic exposure, the cause and effect may not be clearly apparent. When we are exposed to a compound, we will first absorb it into our bodies. Then the compound is distributed throughout our body by our circulatory system. Finally, what is not metabolized will be eliminated. These three processes, absorption, distribution, and elimination, are key processes toxicologists study as they can affect how we respond to exposure to compounds. We can be exposed to a compound through inhalation, ingestion, or absorption. Of the three methods, ingestion and inhalation are the most common routes of exposure. Environmental toxicologists seek to understand the effects of toxic ants and toxins on organisms in their natural environment. The efforts of these scientists can help conserve wild populations and shed light on the risks certain compounds pose to human health. Both fieldwork and lab studies are used in environmental toxicology and wildlife studies. For example, the picture on the left shows a famous wildlife toxicologist, Louise Gillette, taking blood samples from an alligator in a Florida lake. Dr. Gillette was comparing the blood of alligators that lived in lakes that received agricultural runoff to alligators that lived in lakes that did not. He hypothesized that chemical contaminants were disrupting the endocrine system of alligators during development and egg. He later provided with lab experiments that the herbicide atrazine was the source of problems and was indeed disrobing the endocrine system of the reptiles. Building on Gillette's work, Tyrone Hayes found in lab experiments that male frogs raised in water with very low levels of atrazine became feminized and hermaphroditic. Furthermore, field studies showed frogs in areas with heavy atrazine use have hormonal problems. To study human health, scientists use epidemiological studies. To do these studies, epidemiologists compare groups of people that were exposed to a hazard to groups that were not exposed and do so on a large scale, i.e. beyond just a few individuals. Epidemiological studies can last for years or decades. If rates of death, cancer, etc., are higher in the exposed group, the hazard may be responsible. Epidemiological studies show correlation between a proposed hazard and an observed health effect. They do not show causation, meaning a definitive cause and effect. Manipulative experiments, on the other hand, show causation, but these are unethical in humans. Case histories are individual cases of disease in individual patients. While helpful to some degree because they educate us about human illness, they are not all that effective at helping us understand chronic effects or the probability and risk associated with specific disease. Therefore, mammals with shared evolutionary history, such as rats and mice, are used to study substances that may harm them and us. While there are ethical concerns around animal testing, such testing has saved millions of lives of humans. If animal testing is disturbing to you, take heart to know that scientists are developing ways to test chemicals without the use of a live animal, but rather with tissues grown in a lab that simulates a live animal and with computer models. This is still a very developing subfield of toxicology, but it is on the horizon. Toxicologists use dose-response analysis to quantify a substance's toxicity. Dose-response analysis measures the strength of toxic ants' effects by quantifying the number of animals affected at different doses. You should know that a dose is defined as the amount of substance the test animal receives, or the amount of a compound someone is exposed to. A response is the type or magnitude of negative effects caused by exposure to a dose. A dose-response curve is the dose plotted against the response on a graph. In the bottom right you see a dose-response curve following a normal bell curve. This shows that there are some individuals that are extremely sensitive to a dose and some individuals that are very insensitive, but the vast majority of individuals will fall in between these two categories. An important measure of a toxicity test is the lethal dose 50 or the effective dose 50, LD50 or ED50. This is the amount of toxic ant required to kill or affect 50% of the subjects in the study. As you can see in the graph in the upper right, dose is plotted on the x-axis and response, in terms of percent of population killed by the dose given, is on the y-axis. A higher LD or 80-50 indicates low toxicity. Take a minute and look at the graph to process that and make sure you follow why this is the case. If you have questions about this, please email and ask. Toxicologists are often interested in synergistic effects, or the effects that mixes of chemicals have. A synergistic effect means that the mix of chemical may be more toxic than the sum of its parts, meaning that the chemicals magnify the effect of each other when combined together. For example, DDE may help cause or may inhibit sex reversal, depending on the presence of other chemicals. The problem is that we can't determine the impact of all mixed hazards and that they may act in ways that cannot be predicted from the effects of each in isolation. Mixed toxic ants can sum, cancel out, or multiply each other's effects. Again, if they multiply each other's effects, this is called a synergistic effect. When looking at the dose-response curve, toxicologists are also interested in the threshold dose or the level where response occurs. Looking on a graph, the threshold would be where the line changes direction rapidly. You can see this clearly on the graph in the upper right-hand corner of this slide. Organs can metabolize or excrete low doses of a toxic ant. Sometimes a response decreases as the dose increases. This is seen in the dose-response curve in the center bottom of this slide. Here you can see an inverted U-shaped curve. In this case, the dose does not make the poison. These curves are counterintuitive and have been a surprise to encounter in toxicology studies in the past few decades. Endocrine disruptors are a type of toxic ant that display this type of dose response. We will discuss these types of compounds shortly. There are several different types of toxic ants. We will consider 7 in the next 3 slides. 1. Carcinogens are compounds that cause cancer. These can be hard to identify because of the long lag time between exposure and onset of cancer. 2. Mutagens are compounds that cause DNA mutations. These mutations can lead to cancer or they can harm sperm or egg cells and therefore offspring. 3. Teratogens are compounds that cause birth defects to embryos and fetuses. A famous example of a teratogen is thalidomide. This drug was given to expectant mothers in the 1950s as a sleeping pill and a treatment for nausea. Sadly, when the children of women who took this drug were born, it was quickly discovered that the drug caused birth defects and sexual dysfunction in the children. Many of the children born to mothers who took thalidomide were unable to have their own children. 4. Neurotoxins affect the nervous system. Examples include venoms, lead, mercury, and some pesticides. For example, people living on Minamata Bay in China between 1930 and 1960 were exposed to mercury because the nearby plant dumped it into the bay. The people ate the fish in the bay and drank the water. The results of this exposure were slurred speech, loss of muscle control, and often death. 5. Allergens are compounds which cause the immune system to overactivate and provoke an immune response when one isn't necessary. Allergens not technically considered toxins because they only affect some people. 6. Pathway inhibitors are compounds which interrupt biochemical processes in organism. An example would be rat poison. This poison causes internal hemorrhaging and stops blood clotting. The side effect of this is that while dying, the organism is very thirsty and will seek out sources of water. If you use rat poison in your house, 1. Be very careful you, your children, your pets, and non-target organisms are not exposed, and 2. Be prepared for a smelly house if the rat stays in the house, typically the spaces between walls, looking for water. Finally, the last toxic ant type we will consider is endocrine disruptors. These compounds disrupt the endocrine system, which is the hormone system. Endocrine disrupting compounds are called EDCs for short. Hormones are chemical messengers that stimulate growth, development, and sexual maturity. Hormones regulate brain function, appetite, sex drive, and many other body processes. Synthetic chemicals can block or mimic hormones, Bisphenol A and phthalates are two examples of endocrine disruptors. These chemicals can block or mimic the normal hormone signal, thereby disrupting the cascade of effects hormones naturally cause in the body. Because one hormone can have multiple effects in the body, EDCs can cause multiple effects as well. They can be carcinogenic and mutagenic as well. Endocrine disruptions pose challenges because they have unconventional dose-response curves, and these compounds are hard to study because they cause effects at extremely low doses, and it's suspected that they have chronic effects. Therefore, it's hard to set safety standards for these substances. What toxicologists have learned about EDCs means that we may have underestimated effects of other chemicals because other chemicals may also have low-dose effects. In 1996, Theo Colburn et al. Published Our Stolen Future. This book did the same thing for public awareness of endocrine disruptors that Silent Spring did for pesticides. Thousands of studies have linked hundreds of chemicals to effects on reproduction, development, immune function, brain and nervous system function, and other hormone-driven processes. Some researchers say EDCs may be responsible for rise testicular cancer, undescended testes, male genital birth defects, rise in breast cancer, and other processes influenced by endocrine system. BPA is found in hundreds of products. It is in the resins that line food and beverage cans and water supply pipes, and it is in utensils, baby bottles, laptops, toys, and many other products. BPA leaches from products into air, food, water, soil, and dust. BPA linked to cancer, thyroid problems, liver problems, and miscarriages, and is suspected to cause effects at extremely low doses. It is estimated that 93% of Americans have it in their bodies due to continual exposure. Bisphenol A mimics estrogen and as such is an endocrine disrupting chemical. Researchers, doctors, and consumer advocates want governmental regulation on BPA. The chemical industry insists it is safe, but some countries and states have banned it. There is a full ban in Canada, and it is banned from being in baby bottles in the US and the European Union. Many companies are voluntarily removing it from their products. Researchers found that most of these products shown here had leached BPA from the container linings. In regard to BPA research, almost without exception, the studies reporting harm received public government funding, whereas those reporting no harm were funded by industry. That concludes the first mini lecture on environmental health and toxicology. If you haven't already done so, please watch part 2. Please review the weekly outline and complete the activities for this week per the schedule set forth in the outline.